Our first speaker is Arima Genomics' very own Senior VP of Science, Dr. Anthony Schmitt. Without taking too much time here to evangelize and appreciate what Anthony means to Arima, I'll just say a few words. He's the lead developer of the extremely impactful Arima-HiC kit. He's the lead scientist behind the Arima-HiChIP and Capture-HiC solutions. He's well-trained, he's well-published in the field, and he's genuinely a good person to work with. Anthony, thank you and welcome.
Thanks, Joe. Can you hear me well? The connectivity is good?
Yep, we got you.
Perfect, thanks. Well, I'm super excited to be here today. I'm even more excited, I think, to hear some of the published and unpublished work that we'll hear about over the course of the next couple hours. I think my function here is really just to introduce the underlying NGS-based approaches for 3D genome mapping and genome structure analysis, and I'll go over a few of those right now. I'm going to first introduce the 3D genome very, very briefly. I know that we'll hear a lot more about that in the talks to follow. I'll talk about our three solutions for genome-wide HiC analysis there at the top, as well as a couple targeted versions that we've recently come out with called Capture-HiC and HiChIP, and I think each of those are really well suited to address specific targeted hypotheses when it comes to the 3D genome and epigenetics and its relevance in cancer research. So without further ado, I'll get started with the top one there. So first, just a really quick introduction to the 3D genome. This is from a review that my former mentor, Bing Ren, wrote with a couple colleagues in the Ren lab when I was doing my doctoral training there. Here, the idea is just really at the highest sort of level. If you look down at an interphase nucleus, you observe the presence of these chromosome territories, where if you can see my mouse, they're kind of up here depicted as different colors. This is looking down at an interphase nucleus. And this is supported by microscopy data. When you paint different chromosomes, different colors, you see them organizing into their own sort of spatial confines within the nucleus. And then this is also supported by HiC data, where the most number of reads or 3D interactions are occurring here across the diagonal in a genome-wide HiC matrix. So the majority of the signal is interactions within chromosomes, which supports this idea of chromosome territories. And if you zoom in maybe a little bit further, you see other structural features like parts of the genome or chromosomes tending to associate with the nuclear lamina and other parts of the chromosomes extending more towards the nuclear interior. You see the presence of transcription factories and other puncta that are structurally significant features of genome organization. If you zoom in further on the scale, let's say of one chromosome looking at 1 megabase sort of units, you see these self-interacting chromatin domains here that might be called topological domains, TADs, contact domains. There's a few different terms for them that are somewhat used interchangeably. And these are self-interacting structures that are demarcated by architectural proteins at the boundaries like CTCF. And then when you zoom into kind of on the tens to hundreds of KB scale, you see these chromatin loops depicted here with these frequent point-to-point interactions really at the anchor of that loop that might be mediated by architectural proteins like cohesin, and they can also bring together cis-regulatory elements like promoters and enhancers and the like. And this concept of topological domains, I guess, is illustrated here in a schematic where you have a linear thread and you have 2 points, A and B, on that linear thread. Now, if the genome was folded randomly and you were to just crumple up this string into a kind of like a ball of yarn, A and B, which are close together in the linear sequence, would also be close together in the 3D space of the genome. But in fact, the folding of the genome is not random. And you'll see cases like this where A and B are actually spatially segregated despite being relatively close along the linear genome. And these self-interacting domains that are depicted here as this model are also often depicted as these triangular structures when you represent HiC data. And what does that data look like? When you look at HiC data here, this is about a 7 megabase region, this sort of self-interacting domain that I'm highlighting with my mouse would be one contact domain, and then a neighboring contact domain here. And then when you zoom in, like I said, onto sort of a whole nother log order increase in resolution, you see these dots on the Hi-C map, which themselves represent very frequent point-to-point interactions between two regions along the linear genome. So why study 3D genome organization? We're gonna hear some really exciting stories here in the near future, but it's been well, I would say very well studied over the past 10 years or so, just using Hi-C type of techniques alone, but way before that. A couple examples here would be in the context of development. Here's a study by Bing Ren and human ESL differentiation using Genome-wide Hi-C. In the context of immunology by one of the speakers here today. In the context of neurobiology from the Akbarian lab at Mount Sinai, and then also in the context of cancer, which we'll hear more about today. And here's a couple studies also from our speakers today, and I think we'll probably hear more about these later on. So getting to the underlying tools, which is what I'm going to introduce. When we talk about the Arima-HiC platform, let's start with the Genome-wide Arima-HiC Foundation. And it frequently comes up, what are the supported sample types? And there's a lot of supported sample sources shown on the left. I think the most relevant for today is going to be human and mouse. There are another suite of sample sources that are supported. In terms of the sample types, we have user guides for cultured cells, primary cells, whole blood, fresh or frozen tissue. We've worked with OCT embedded tissue. So if you have a specific question about a sample type, please drop that in the Q&A or we can follow up afterwards. And then in terms of the sample amounts, a typical Arima-HiC reaction might start with about 500,000 to 1 million cells. If you're working with frozen tumor tissue, you might start with maybe 50 to 100 milligrams of that tissue, grind it up, cross-link it, and then probably use a fraction of that for your Hi-C experiment. And then if you're working with, let's say, mouse blood, you might collect 2 to 3 mils of blood, isolate PBMCs or some type of subpopulation from that, and do your Hi-C's experiment on that. And I should also point out that we do have low input protocols available, and I don't have time to go through our low input Hi-C internal data today, but I will point you to one paper that's used our low input Hi-C. Now, really to get into the nuts and bolts here of the workflow, if anyone, if you're familiar with, let's say, ChIP-seq, Hi-C starts in a very similar fashion. You start with your fixed cellular input here, and then while the chromatin is held fixed there within the nucleus, there's really 3 key enzymatic steps that are carried out. First, we digest the chromatin with a cocktail of restriction enzymes. So what is depicted here is essentially 2 pieces of DNA when you're looking down on a cell that are not close along the linear genome, but they are held together in 3-dimensional space by this protein. Once you have your digestion here, those ends get labeled with a biotinylated nucleotide and then ligated back together. So what you started with was 2 pieces of DNA that were just held together in 3-dimensional space, but not actually physically adjoined to each other, now end up cut and religated back to each other. And you have biotin marking the ligation junction point between those 2 pieces of DNA. And this is the most information-rich piece of this molecule where these 2 molecules ligated together. And then you just purify that DNA and you do a quality control checkpoint that we've built into the technology here that is predictive of the quality of the final sequencing data. So it's an experimental predictor of success. Now, I think I've gone through here the sample input requirements. This whole process, which we refer to as Hi-C, is completed in 8 simple steps. The entire process happens in a single tube. You just keep piling on reagents from the minute that you lyse your cells through the final purification of DNA, which is quite nice that there's no spinning or anything that would be susceptible to sample loss. If you're familiar with other Hi-C protocols, we've innovated in using a cocktail of restriction enzymes, which increased the theoretical resolution of the assay and also the coverage uniformity across the genome. This entire process takes about 6 hours, or about one working day to do the Hi-C part. And like I mentioned, this is a predictive QC checkpoint. Now, after this, these molecules here that you purify are a little large. They're about 3 kb. So we shear those molecules, and then we use streptavidin beads to purify the part gray and part blue molecules, which really capture and represent that three-dimensional interaction that was occurring within the nucleus. And then we wash everything else away. We add our sequencing adapters using commercial library prep kits like Kappa, Swift, NEB, Illumina, etc. And then you amplify, and then you sequence these chimeric molecules, and then you map those back to the reference genome. So that will be essentially one data point that, let's say, chromosome 1 and chromosome 4 were interacting within 3D space. I did mention that we have low input protocols. We've gone down to 50,000 cells or less. I would just point you to a recent publication there from the Akbarian lab that I mentioned earlier that had used our kit for Hi-C analysis of 6,000 sorted dopaminergic nuclei that were harvested from postmortem brain samples. And we offer this technology as both a kit and a service format. And I really only have time to show, I think, one piece of data to highlight the benefits of using the kit. And that is really when we first started generating this Arima-HiC data, we of course set out to benchmark that data against really the gold standard in situ Hi-C data that was available at the time. And that was really an excellent protocol and has continued to be a workhorse in the field. So when we did this comparison, we took some published in situ Hi-C data from 3.6 billion raw Hi-C reads. And all we're looking at here is essentially one point-to-point interaction, which would be characterized as a chromatin loop, about a megabase in size. And when we analyzed the same sample using the Arima-HiC data, what we observed is that at these looping regions, essentially these structural features that you're often doing Hi-C to detect, that signal enrichment at that looping region was much higher than in the published in situ Hi-C data. And I think the important thing to notice is that signal is more enriched despite there being only a third of the total sequencing depth. So the concept here is just that really in this case in point example, we're highlighting that the signal strength of these structural features is higher despite having less reads than other Hi-C methods. And to take this one step further, what we did is we took the 1.2 billion raw read Arima-HiC data set and we subsampled that down to 1 billion reads, 800 million reads, 600 million reads, and we called loops using the open source tools called Juicer and HiCups. And I'm just plotting the total number of loops that we find compared to the 3.6 billion read published in situ Hi-C data set. And what we find is just that the number of loops that you can detect is about 50% higher, despite having a sixth of the total sequencing depth. And that's internal data, and our customers have also seen this as well in a different cell line. Again, the concept of being able to detect more loops from less sequencing depth. And there's been several testimonials of those who have used our kit in the field. This is all on our website, and a growing list of publications in the nuclear architecture and epigenetics research fields, which are also on our website. Now, with that introduction really to the Genome-wide Hi-C, as a company, we've been really, I think, keen to address more targeted tools that are well positioned to address specific targeted hypotheses when it comes to genome organization, gene regulation, and those two tools, like I said, are called HiChIP and Capture-HiC. And I really only have a time to just barely touch on both of them. And I'll start with Capture-HiC. I think a good way to introduce Capture-HiC to those who are not familiar is to position it relative to more popular capture sequencing type of protocols like exome sequencing. So in a typical exome sequencing workflow, you extract your DNA, you shear that DNA, you add your sequencing adapters, and you amplify. And then at that point, before you would sequence, you introduce these biotinylated oligonucleotides that hybridize to the exome, for example, and then you can capture those hybridized molecules using streptavidin beads and enrich your genomic DNA library for the sequences that you want to capture, like the exome. Then you amplify and then you sequence. So that's a typical capture sequencing protocol. Now, the way that Arima Capture-HiC is different is this process of extracting the DNA is simply replaced with doing Hi-C. So instead of just extracting DNA, you go through the process of digestion, biotin fill-in, proximity ligation, and then DNA purification. Right. So in fact, really, it's the exact same Hi-C protocol that you would do if you were doing genome-wide Hi-C, except right before you would get to the point where you would traditionally sequence, then you add your biotinylated probes and you enrich for any sequence element of interest that you're interested in probing for the 3D landscape. So you can imagine if you want to capture promoters and all of the distal elements that interact with promoters, you would do an experiment where you enrich the Hi-C library for promoter sequences. And we've been partnering with Agilent Technologies over the past year to try to streamline this Hi-C plus capture sequencing protocol, and been using their probes, hybridization reagents, and probe design software suite. So really, I think just to get you thinking about how this might be useful in your research, there's a couple different flavors, I would say, of Capture-HiC to think about. There's the concept of, let's say you have a gene or several genes of interest, and you're interested in obtaining Hi-C type of data around just those genes and not the whole genome. Then you might do an experiment like region Capture-HiC, where you design your capture probes to enrich for the contact domain that contains your gene of interest and maybe the flanking domains as well, but then leave everything else behind. There's also been a body of literature that has used Capture-HiC with the concept being there's lots and lots of GWAS studies that have identified variants that are associated with a disease phenotype, and perhaps those causative variants in the non-coding regions are disrupting, let's say, enhancers, and you want to use the Capture-HiC data to link those regions to putative target genes that may be associated with the disease phenotype. And lastly, like I said, the concept of promoter Capture-HiC, where you capture promoters and everything that interacts with those promoters, either at a genome-wide scale or some type of subset of all the promoters in the genome. And the way that we work with our customers for Capture-HiC is we help really through the entire process of designing the probes. We use the Agilent Sure Design software suite. We have off-the-shelf promoter Capture-HiC designs for mouse and human. We also do lots of custom designs. We review the design with you on the UCSC genome browser, and we iterate if necessary, and then those probes would be ordered from Agilent. Now, we have a lot of really exciting, I think, internal data. One of those experiments that we've done internally is we've captured the regions around several different select oncogenes or tumor suppressors, like MYC, EGFR, BCR, PTEN, NTRK. And we've done this in a way where, let's say, I'm saying here we capture BCR, 620 kb. What I mean is if the BCR gene is here, we've designed probes to capture the 620 kb around the BCR gene and obtain that high resolution, Hi-C data just around BCR. And we've done this in 3 different normal cell lines and seven different cancer cell lines of different tissue origins. All this data that I'm going to talk about here in the future is openly available if you want to have a look at it. So what does this data look like? So this is the K562 Capture-HiC data. And I want to step through, I think, the real benefit of an experiment like region Capture-HiC. So let's look at the BCR gene. if you were to take this Capture-HiC data and analyze it at a large bin size, let's say 25 KB bin size, you would get, something like this, where you have an interaction that you can kind of see that's happening off the diagonal here. And when you trace those back to where those originate along the linear genome, you can see that they're here. It looks like a very pixelated image, and you can't really tell when you look at the chromatin features whether it's CTCF peak here or here or here or here that's associated with this interaction. And you might really want to know whether that interaction is at the promoter of BCR, but you can't resolve that at lower resolution. You can take the same Capture-HiC data and bin it at 5 KB resolution, but unfortunately, you actually still run into the same issue where if you look at that significant interaction that appears to be happening, and you trace that back to where it belongs along the linear genome, again, you can't really tell if that loop is at the promoter of BCR or just upstream of BCR. You can take the same Capture-HiC data and cut the bin size by a whole nother log order down to 500 base pair resolution, and now it becomes much more clear that that interaction that you saw when you trace that back to the linear genome is really anchored at the promoter of BCR. So this type of 500 base pair bin size is really unprecedented at the genome-wide scale. It would take on the order of probably, 8 or 10 billion reads genome-wide HiC to get this resolution. But you can get this type of resolution from targeted HiC around your gene of interest for approximately 8 or 10 million reads per locus like this. So for Capture-HiC, just in summary, we've developed a streamlined Capture-HiC protocol that combines Arima-HiC with Agilent SureSelect. We have a full support, completely complementary for probe design and experimental consultation. And here I've showed you how you can get sub-kilobase, ultra-high resolution targeted HiC maps around your genes of interest for approximately 10 million total raw reads per megabase of targeted sequence. Now, lastly, we'll go into HiChIP. Similar to how I introduced Capture-HiC, to introduce HiChIP first, in a typical ChIP-seq type of experiment, you have your cross-linked cells, you shear that chromatin, you add your antibody, you do your IP, shown here, and then you do your library prep and sequence. Arima-HiChIP is very similar in that it's actually just as it sounds in the name. You start with an upfront HiC, so again, digestion, fill-in, ligation, and really at this point, you have this proximally ligated chromatin. Then you shear this chromatin and you add your antibody. So when you structurally look at the molecules that are getting enriched, the difference is that the HiChIP ones, this molecule not only contains the sequence content around the immunoprecipitated protein that you would get from ChIP-seq, but it also contains a 3D interaction information that was associated with this immunoprecipitated protein. So after you do your IP here, then you do your HiC style library prep to enrich for the ligation junctions. So in this workflow, there's essentially two enrichments, one for the protein and then one for the ligation junctions. And this technology inherently measures the 1D protein localization sites that you would get from ChIP-seq, and then also the 3D interactions that are associated with that immunoprecipitated protein. And lots of the customers so far are using this technology as a tool to characterize chromatin loops that are associated with active promoters or active enhancers. We also have optimized sharing protocols that are compatible with Diagenode as well as Covaris. We've had a really fruitful collaboration with Covaris developing the shearing component of this protocol, and I would just point you to the poster that we presented with Covaris at AGBT this February. And we also offer this as kits or services. Now, why use HiChIP? It's been used in the context of cancer epigenetics a couple times here by the Bernstein lab and Paul Mischel's lab here at UCSD. I would just point you to those papers. when we talk about visualizing the Arima-HiChIP data, this is a WashU epigenome browser track around the MIC locus here. So we have our CTCF ChIP-seq data, cohesin ChIP-seq data, et cetera. What I wanted to draw your attention to here when it comes to the performance of Arima-HiChIP is the bottom two tracks here are essentially two replicates of ChIP-seq data for H3K27 acetylation downloaded from ENCODE. When you plot the HiChIP data in a 1D signal track, just like ChIP-seq data, the 1D protein localization sort of occupancy data, that 1D signal track in both replicates recapitulates the ChIP-seq data. So you're still capturing those same ChIP-seq peak information around the MIC locus. And it's very similar compared to ChIP-seq. But what you're not getting from the ChIP-seq is that 3D connectivity information that connects these K27 acetylation peaks to other K27 acetylation peaks or promoters or other cis-regulatory elements in the locus that would inform you of a 3D gene regulatory type of interaction. And we've also tried to be thoughtful about making sure that this technology is reproducible in terms of the loops that it calls. So these statistically significant loops that are being identified here using the open source MAPS pipeline appear to be quite reproducible at this locus. And at a genome-wide scale, we find that about 85% of the loops that we find in Rep1 are also found in Rep2 for the K27 acetylation HiChIP, and a similar degree of reproducibility in our H3K4 trimethylation HiChIP. So the last slide I have here is, I think Arima really prides itself on putting a product out in the market that's been customer validated. And I wanted to go through some of the beta testing data that we've completed before launching the product. So just a couple key performance metrics when it comes to HiChIP. You really want to query the data to make sure that the HiChIP data has a high efficiency of proximity ligation. In other words, it has a strong high C signal in the data, but it also needs to have a high chip specificity signal in the data. So really with high C, you're just looking at what are the fraction of long range interactions that are in the data, and with chip, essentially what fraction of the reads overlap known chip peaks. So when you plot that data, the percentage of long range interactions in the HiChIP data on the y-axis and the fraction of HiChIP reads that overlap chip peaks, you want to be in the upper right quadrant of this plot. And all 24 reactions from the 3 beta testing sites meet that criteria of having a high fraction of long range interactions and a high chip enrichment for both antibodies. We then plot essentially what are the total number of reads that are required for at least 70% reproducible loops across the 3 different beta testing sites in many, many different cell lines that were examined across these 3 different sites. And the main takeaway here is that for H3K4 trimethylation, we're needing about 100 million reads to obtain reproducible active promoter-centered loops across the genome. And for K27 acetylation, a bit more of an enhancer-centric point of view, about 300 million reads for those H3K27 acetylation-mediated loops. And if you step back and just sort of think about the sequencing requirements for these types of technologies, for in situ high C, for loop level resolution, I believe ENCODE is recommending about 2 billion reads. For Arima-HiC loop level resolution, we're talking about 600 million reads. And then for the HiChIP, it's about 300 million reads for K27 acetylation and 100 million reads for K4 trimethylation. I would just lastly direct you to some of the testimonials from these early access partners and beta testers. And in conclusion, for HiChIP, I think we've developed a very nice streamlined in situ HiChIP type of protocol for the efficient capture of both the 3D loops and the 1D chromatin occupancy signal. Those loops are quite reproducible, about 80%. They've been validated across multiple protein factors and cell types. And I didn't have a chance to talk to this, though I'm happy to answer any questions about this, is we have validated multiple open source HiChIP analysis tools, and all this is on GitHub, and we provide extensive technical support, training, and onboarding for the bioinformatics piece. So with that, I'd just like to acknowledge our scientific advisors, Job Decker, Jesse Dixon, and Bing Ren, and the funding support that we've received from the NIH to develop the tools that you heard about today. And if I have any time, I can take a couple questions. Thanks.
Great. Well, thank you so much, Anthony. This is Joe again. Just to keep, in the interest of time, let's keep this moving, but let's take a couple. How about the very first one? Can we use Arima kit on more than 1 million cells? You mentioned the low input capability, but what's the upper level?
Sure. So the upper limit we recommend for the genome-wide Arima-HiC or Arima Capture-HiC, it's actually really limited by the amount of chromatin that's in the cells that you put into the assay, because effectively the chromatin that's within the nuclei is the substrate to these enzymatic processes. And we try to work with about 5 micrograms of chromatin within those cells or less. It certainly doesn't mean that using more cells will fail, but the number one cause that we've seen when we try to break the experiment or break the assay is if you put in too many cells, the biochemistry does become inefficient and doesn't run to completion. So I think in short, yeah, we try to aim for, usually it's about no more than 1 to 2 million cells, and then it can definitely go down from there, and sometimes less is more.
Less is more. Great ending. Yeah, that's a helpful hint for those using the kit. Thanks a lot, Anthony.